Hello, everyone, and welcome back to the NWR Virtual Healthcare Day for November 2021. My name is Finn Blake, and I will be your host for this afternoon. Up next this afternoon, we have ImmVirX. ImmVirX is developing novel oncolytic viruses to create powerful new cancer immunotherapy combinations. The company was founded in 2019 by the team at Viralytics, an ASX-listed biotech which was taken over by Merck in 2018 for $502 million. ImmVirX recently closed out a $25 million Series A financing round led by Acorn Capital and several other well-known local institutional investors, providing a cash runway for the company through until the second half of calendar year 2023. Joining me this afternoon is the Interim Chair and CEO, Dr. Malcolm McColl. Thanks so much for joining us, Malcolm. And if anyone in the audience would like to submit a question for the Q&A session, which will happen at the end of the presentation, then please don't hesitate to type it using the Q&A panel on your screen. Do that anytime throughout the presentation and we'll get to your question at the end. Over to you, Robert. Over to you, Malcolm, sorry.
No worries, Finn. Thanks so much. And look, thanks also for the invitation to your NWR Healthcare Day. As you said, ImmVirX is a company that's developing oncolytic viruses for difficult to treat cancers, and we're excited to provide an update and also look forward to some pretty important milestones that are just on the horizon for us. So perhaps just the next slide. So this is just our disclaimer, and I'll refer you to our website to see that. Look, I think folks who are familiar with the Viralytics story will recognise many of the names and faces on this particular slide. Obviously, Darren Schaffman and myself. Darren was the inventor of Viralytics Cavitac, CSO from Viralytics. I was the CEO, so we formed ImmVirX in late 2019. Then joined by Robert Vickery, who is currently our part-time CFO, also formerly of Viralytics. Delighted to have Len Post, who was a very important director at Viralytics, a highly regarded and respected biotech executive out of the Bay Area in San Francisco, coming back on board. Robert Routley, CEO of Acorn, is joining us as a non-executive director. Acorn led the Series A financing, successful Series A financing we've recently completed, and Jen Rosenthal was our Director of Quality and Reg Affairs at Viralytics and has taken on the same role at ImmVirX. So what you have really here is the leadership and the scientific team of Viralytics. This is the team responsible not just for the invention of Cavitac, but also taking it through preclinical, early stage manufacture, scaling that up to large scale manufacture, then through to the clinical development and ultimately through to acquisition by Merck for $500 million in mid 2018. It's a team with deep regulatory knowledge, lots of face-to-face interactions with FDA and other regulatory agencies, a really deep experience in terms of manufacture and quality systems. That experience was further honed. Many of the team stayed at Merck for a further 12 to up to 18 months after the acquisition and learned a lot more through that period. We have a great global network of world-leading key opinion leaders and oncologists who help us think about the clinical program moving forward at ImmVirX. And again, the R&D team, really the heart and soul of the business. We're delighted that we've been able to reform that team with all the important team members from Viralytics and with some new folk we've recruited as well. And again, delighted to work again in the same facilities as Viralytics at the Hunter Medical Research Institute in conjunction with the University of Newcastle. And as Finn said, just completed successful financing of $25 million that takes us through to the second half of calendar 2023. So look, this is an important space. Oncolytic viruses, we think, will be a very important space in this whole field of cancer immunotherapy. I think most of the audience are probably familiar with cancer immunotherapy. It's the largest part of the global pharmaceutical industry. Really, and more importantly than that, it's really transformed the way we're able to treat cancer patients. And in many cancer types, we've been able to see long-term survival benefits for patients, particularly in indications like lung and melanoma and bladder. But regrettably, still only a subset or a minority of patients are actually successfully treated with checkpoints. And so there's a race in big pharma and biotechs around the world to find agents to combine with checkpoints to make them work better, work in indications where they're currently unsuccessful. Merck's acquisition of Viralytics was part of that general thrust to find new agents. And we think that ImmVirX has a similar prospects moving forward. Despite those sort of limitations, it's still a massive space. Annual sales are projected to be 125 billion US by 2024. They're currently around about 80 billion. So we're seeing double-digit growth in this space. And for reasons I'll describe in a bit more detail in a minute, we think oncolytic viruses are a great combination with checkpoints and other cancer immunotherapies to make them work better. Obviously, with this sort of level of potential, there has been activity from big pharma in terms of acquiring companies. A couple of noteworthy ones: Amgen's acquisition of ImmVirX for 425 million upfront, Merck's acquisition of Viralytics for 502 million Aussie upfront, and then also a lot of interest from capital markets. The best example on NASDAQ, I think, is Replimune. This is a company that was formed by the team from ImmVirX in around about 2015. They did their IPO in 2018 at around about a 460 million valuation, and today trading around about that 1.5 to 1.6 billion market cap as they advance their program in the clinic, particularly in types of skin cancer. Next slide, please. So I think when you start looking at indications where these checkpoint drugs like Keytruda from Merck aren't really very successful, you look at ovarian, colorectal, gastric, and pancreatic cancer, where response rates, as you can see in the fourth column there, are either in the single digits or sort of mid-teens. So really lots of room for improvement there. And then you look at the importance of these cancer types. I mean, colorectal cancer is the #2 killer in terms of cancer deaths in the US. Ovarian and gastric and pancreatic also very significant, and all of these really much greater than melanoma, which was the primary target of Viralytics. So if we have success in this sort of setting, obviously the valuation of ImmVirX could be some multiple of what we achieved with Viralytics. Looking at China, I think you can see also there another really important market, and colorectal and gastric cancer are the #2 and #3 cancer types in that country. So these are cancers that are very common. There's a really high unmet need for better therapies, and this is the area that we're targeting at ImmVirX. Next slide, please. So our platform at ImmVirX is to, again, take a small RNA virus within the same broad family as Viralytics Cavitac. Obviously, it's a different virus to Cavitac, and we put that through a second generation or a next generation bioselection process that enables the bioselected virus to preferentially target receptors that are overexpressed on the cancer types that we have just spoken about. This bioselection process then allows the virus to really exquisitely target, select out these cancer cells, infect those cancer cells. We know the virus then can multiply very rapidly and kill those cancer cells. So it's a pathway. You'll see a little data on that in a minute as we look at some of the preclinical program. It's a proven mechanism. I mean, these types of small RNA viruses we saw with Viralytics Cavitac were able to really effectively infect melanoma tumors. As they infect, they really drive a strong immune response. The immune system sees this replicating or multiplying RNA virus as foreign, so it draws in lots of immune cells. In the meantime, the cancer's being destroyed, being lysed, and so fragments of the cancer cell are then exposed to the immune system. That then gives the possibility of these immune cells or T cells then migrating around the body to attack cancer, not just at the injected site, but at other parts of the body. And we certainly saw that with Viralytics Cavitac, where we saw wonderful activity in liver and lung lesions and at other sites beyond injected lesions. And we'll speak to a minute about the preclinical program and some of the proof of concept data that we have at this point. The clinical strategy really is to look at colorectal, gastric, and pancreatic cancer, initially as monotherapy, and then we'll move forward with combination studies in combination with immune checkpoint inhibitors. And we have a clear pathway to trial initiation expected in mid-2022. Next slide, please. So our lead candidate here is IVX-037, and we'll take a look now at some of the preclinical data. The studies here are all pretty similar in terms of the way we're tackling this. Pretty standard sort of model. We use immunocompromised mice or mice that really don't have an effective or operational immune system. We implant the tumor cells on the left flank, and then after a period of about 2 weeks, we inject those tumors either with IVX-037 or with a vehicle that does not contain the virus, and then we monitor those mice over the next 2 or 3 weeks for the size of the tumor and also how well tolerated it is, the health of the mice essentially. Next slide, please. The first of the models we looked at was in a human colorectal cancer cell line. You can see there on the graph on the left-hand side, the red line is the sort of mean tumor size in the mice following treatment with IVX-037, and the blue line is the control group. So you can see a distinct sort of separation of the curves there and really excellent activity. Again, here, this was just a single dose of IVX-037, so we saw quite nice sustained activity, and it was also very well tolerated. Next slide, please. We then wanted to take a look at different dose levels. So we kicked off with a dose of only 10,000 virus particles up to 100 million virus particles. You can see those in the red, green and purple lines, as opposed to a vehicle with no virus. And again, this is in this colorectal cancer cell line, and it really didn't have any impact whether we used 10,000 or 100 million virus particles. We saw wonderful suppression of the tumor growth as compared to the vehicle. So I think this is important because it shows there's quite a wide therapeutic window here, or quite a wide range of doses where the virus is active. And in fact, it's active at very low dose. So that's important as we move forward, I think. We then looked at a further colorectal cancer cell line. Again, really nice separation of the curves. Here you can see excellent suppression of the cancer with IVX-037 versus the controls, so confirming activity in colorectal cancer. We then decided to take a look at gastric cancer on the next slide. And in this case, you can see this was a very aggressive, fast-growing gastric cancer cell line, advancing very rapidly in the control group. There is suppression achieved by a single dose of IVX-037 in this case. So again, a really nice result, and again, well tolerated. Again, just to emphasize, just a single dose of virus given in this case. We then decided to switch gears and take a look at ovarian cancer. So again, here a human ovarian cancer model, and you can see again the clear separation between the treated group and the control group that has the vehicle without any virus in it. We then elected to move to a second ovarian cancer model, and again, you can see the same sort of outcome. So it was a nice body of work demonstrating activity across a range of cancer types. We then wanted to take a look at intravenous delivery. So again, a very similar sort of design for the study where we implant the tumors, but then rather than give a single intratumoral dose, we gave 4 doses intravenously and then monitored those tumors over the following 2 or 3 weeks. This is again in colorectal cancer. And again, here we see quite nice separation of the curve, suppression of growth of the tumor by this IV delivery as compared to the vehicle group. So a pleasing result. So I think sort of at the conclusion of this sort of first round of preclinical work, and there's further data that we have that's not in the deck today, I think it's really pleasing that we've seen activity across a range of different cancer types. We've seen it across a range of different doses, including quite low doses. And we've also seen it via different routes of administration, intratumoral and the intravenous administration. So a really excellent package of data, including, very well tolerated in the mice as well. So I think we completed the first tranche of the financing in May of this year. We're able to then recruit some further members to the team, which is exciting. And I just wanted to draw everyone's attention to what's been achieved since that time. I mean, no other team, I think, could have progressed as rapidly as we've done, considering the company was only really put together sort of early in 2020. But even since May of this year, rapid progress. So you can see the results we've achieved across further xenografts in colorectal, gastric and ovarian. We needed a small animal model to enable us to do preclinical tox studies to satisfy FDA and other regulatory authorities. We've imported that mouse model, and that's about to get underway, which is excellent. We've taken a look at different routes of administration, including IV, obviously, and colorectal with ovarian to follow. And, we've done some further kind of very elegant science, actually, to further demonstrate this interaction between the virus and the receptor that is important in terms of proving mechanism of action to big pharma and global institutional investors, and also enables us to broaden out the patent portfolio. So exciting in terms of the preclinical and IP. In terms of manufacture and quality, we had already prepared the master cell bank prior to May. We've since May prepared the master virus seed. These are sort of critical precursors prior to manufacture. They need to be prepared in a GMP facility. We've obviously taken a lot of work's been done to prepare us for Australian production of IVX-037 for the first inpatient studies, and that production is now getting underway. We also plan to manufacture again in the US, and we've signed up a relationship with the company that previously made Cavitac for us in the US, and delighted to have that underway with tech transfer already occurring. And importantly, we've done some work on the viral formulation. The existing FDA approved oncolytic virus TVEC has to be stored at -70. Cavitac is to be stored at -70. And we thought, we had feedback from many that it would be good to have a formulation where there was a slightly more user-friendly storage temperature. And so we've done some nice work actually, and shown early days that 2 to 8 degrees, formulation that's stable at 2 to 8 degrees. So that could be a really big value add as we move forward and talk to pharma partners. And then in terms of in the clinic, the clinical trial protocol is pretty much done. We'll be doing a sort of a standard phase 1A, phase 1B. This will be a basket study. We have Australian key opinion leaders and oncologists identified who will be excited to participate in the phase 1 study. And we have a short list of CROs, and we'll be finalising our selection of CRO actually quite soon. And again, just to emphasise that since May, we've been able to increase the size of the team with some further new hires in Newcastle and also in other parts of the business. So it's been a very sort of successful few months. Next slide, please. In terms of upcoming milestones, we'll see further IV preclinical studies complete. Our preclinical tox will be complete early next year. We are going to look at assessing IVX-037 in further indications, that further cancer types that express this particular receptor. There's many other targets we have actually. We're excited to do that work. And then as we move forward, we'll also do some preclinical work looking at combinations, much as we did with Viralytics, combinations of IVX-037, not just with immune checkpoint inhibitors, but also with CAR T cell therapy. In terms of manufacture and quality, we'll complete production at the Australian sites for the first inpatient studies. Tech transfer is already underway to the USA. Our engineering runs will then commence, and then first GMP batches in the middle of next year. In terms of the clinic, we'll appoint a CRO shortly, then do all the necessary steps to get underway with our first inpatient studies in Australia, in terms of ethics applications, getting agreements with trial sites, ethics and TGA approval, then aiming for our first patient on study middle of next year. And then subsequent to that, look to plan for our US FDA IND filing, which is really would then enable us to conduct studies in the US. Finally, just tracking the progress of the company. We saw June 2018, an exciting time when the acquisition was complete with Merck. Most of the team then stayed for a further 12 to 18 months. Darren and I got back together at the end of 2019, did a seed round, then completed a research services agreement with the University of Newcastle, got underway with the science. The candidate was nominated around about April. In 2020, we put that through the bioselection process, then did small scale manufacture, moved into preclinical proof of concept, early studies, then our Series A, first tranche completed in May and then finalised just recently. Then expanded our preclinical models. You've seen some of the results from that today. Necessary steps in terms of production of the master cell bank and master virus seed stock. Preclinical tox is now the next step, manufacture for phase 1 clinical studies, and then first patient dosed in the phase 1 study middle of next year. We should be looking for some early results from that phase 1 study, sort of later 2022, early 23, and then getting underway with combination studies. So a lot to look forward to. A lot's been achieved with this really outstanding team, particularly in Newcastle, and a lot to look forward as we move forward. So perhaps at that point, Finn, I might just draw it to a close and welcome any feedback or comments from yourself or anyone in the audience.
Yeah, beautiful, Malcolm. Thanks so much for that. I do actually have a couple of questions that have come through here. I'd love to ask, is the plan to definitely list the company or not necessarily, and what will contribute to that decision?
Yeah, look, I think that at the moment we're sort of really just flat out doing all the steps I've just talked about in terms of getting into the clinic. I think the board's taken the view that we kind of get to, March, April next year when we can kind of catch our breath and then just really assess at that point. But really all options are on the table in terms of whether we look to list here or in the US or whether we, move forward in other directions. And in terms of your question, in terms of which pathway we might take, I mean, it's really about, these things are always about just sort of drawing the best possible investors into the company. So how do we bring those best investors? What's the mechanism to bring the best possible investors? Because you want people that are going to support you today, next week, next month, and next year. And so we know what those investors look like. We had them in ImmVirX. And so that's what we sort of, that will really be the driving factor, I think. How do we bring those people on board?
Awesome. One last one from me, Malcolm. What is the main reason that you're confident in the ImmVirX business as we approach the new year?
Well, look, I think it's, the virus IVX-037, we put that through a superior bioselection process. All the sort of preclinical data to this point looks as good or better than we saw with Cavitac in melanoma. So in terms of tackling these tumor types, the data we've generated to this point is really, first rate. The key thing, it's all about, talent at the end of the day. It's all about the team, and having a team that's kind of been there and done that is really absolutely critical. And I'm just, as I said, I'm so delighted that we've been able to reassemble this team. They had the benefit of working with Merck for, periods of 12 to 18 months. So they're even stronger now. They've been through a very comprehensive diligence process during the acquisition period. And so to reassemble that team with the talent they have, just look at what we've achieved in the last sort of 18 months, it's remarkable.
Malcolm, thank you so much for your time today. Really looking forward to seeing what this company has in store.
Thanks so much, Finn. Bye for now.